I'm going to figure out this technology yet, but it's difficult. Uh, Nevertheless, so this morning on the bottom of your blackboard, I put this PowerPoint, what we'll look at today, this biomolecules. And so it's here, and I have it, and I loaded it and opened it up because I figured that a little bit more of the basics wouldn't necessarily hurt before I get over to this one, which is PowerPoint D, which has to do with the cell cycle, which is really perhaps a major topic in this area. So, I mean, this sort of overlaps both things. I mean, I really wanted to do this for DNA, but as I opened it up, I thought it might not hurt to talk a little bit about water in the context of doing this. Because we take it for granted, but we're somewhere, depending on what author you read, between maybe 2/3 to 3/4 water. I guess depending on how well hydrated we are in the morning or in the afternoon or in the evening. Nevertheless, so this is this is the same thing materials we use for the basic anatomy and physiology. It's the same thing I use for the advanced anatomy and physiology classes as well. The same textbook. It's just a matter of how much detail one chooses to put into it. So let us begin. So biochemistry is really the chemistry of living molecules, and it plays a particularly, I think, important role in what we're doing here, or what we're acquainting you in this class, is the effects of radiation. And the two principal areas that are directly water, which I think Dr. Buxton will handle much more of than I will, and DNA, which will be sort of in my bailiwick. But what DNA indirectly, when there's damage to DNA, and damage to DNA, we have a word for it, they're called mutations. And what that does, okay, is it causes alterations in protein sequences. So the direct effect is on the DNA, the indirect effect is what it does to the DNA, causing its message to become erroneous and alter proteins. And as I say 1,000 times, you are what your proteins say you are. So the chemical composition of living matter. So we put, even though inorganic means that it is not composed of carbon compounds that are covalent, we call it covalently bonded carbon to carbon and carbon to hydrogen. The oddball here is carbon dioxide. Okay. Carbon dioxide is covalently bonded, which means a sharing of electrons. But by definition, it's inorganic. That's not a big deal for us, this organic be inorganic or anything along those lines. So, but the organic compounds are 4 basic groups, and I will cursorily look at those 3 and much more detailed look at the nucleic acids, specifically the DNA. So they contain carbon, and again, usually large, covalently bonded, and again, you'll see carbon to carbon and carbon to hydrogen. But being important for living is certainly all part of it. So water. So here you can see, depending on the cell you're dealing with. And water is miraculous. Water is a very interesting combination of identical molecules that have 2 things. And you see the illustration. So if this has the capacity to alter water, water is very, very sort of fascinating. So it's combined of it. You'll see the diagrams. Water is a relatively simple molecule. Everybody knows it's H2O, right? It's got an oxygen with 2 hydrogens, without me trying to draw. I am the world's worst artist. And I can't play the violin either. No musical talent, basically. So, thanks again. Well, the only person who smiles right up there in the middle, whoever you are. The rest of you, big trouble. Big trouble. So you have an oxygen with 2 hydrogens. And what's interesting about that, that's a covalent bond. They're sharing electrons. But because of the positioning of the hydrogens, it is somewhat off kilter. So it lacks a certain degree of, let's call it, electrical symmetry. So that undoubtedly is an influencing factor in some of the things that happen to it. Where typical organic molecules, particularly things like fats, have inherently electrical stability, so they're pretty resistant to what radiation does. So how does a microwave work? Okay, we are able to take what you call polar molecules and basically excite them, get them to move around faster, and they generate heat. That's how a microwave works. So and again, I don't want to go over into something that I have any degree of expertise. But because of that, this sort of asymmetry, they have a sort of an irregularity in charge. And maybe I should show it to you before I be blabbing about it, like this. So it kind of looks a little bit like this. The oxygen, because of this asymmetry, because the hydrogens are on one side, the oxygens are kind of on the other, the oxygen has the ability to pull the electrons closer to it. So it creates a partial degree of negative charge. So kind of like this side of the molecule is kind of negative, this side is kind of positive. And that's what we refer to as polarity. And so that allows, as energy is introduced to it, it tends to move it around irregularly to a certain extent. I mean, it does other things, so it can slide between things like that. These are called ionic bonds, positives and negatives. So it's a crystal of salt, just a little lump of salt. But immediately put it in the water, it dissolves. That's because the water can sneak in between these relatively weak bonds. They're just a magnetic attraction, just positive attracting negative. So water has that ability to dissolve things, but at the same time, it has this remarkable ability, and you saw that previously, okay, because that was the polar solvent property. And the fact, and just the bulk of the water, you can understand sort of cushioning, but the reactivity is something I think they'll explain with the radiation. But this, heat capacity and heat of vaporization have to do with another principle, okay? And I want to get to where that is. Not the acids and bases and things like that. Oh, heavens. It's not on this PowerPoint. I didn't write it, but okay. What water does, so if you have a bunch of these water molecules, they're not individuals. They have tiny magnetic bonds in between them. And the reason I wanted to bring this up to you is this has a lot to do with the deal. Something called a hydrogen bond. So water molecules, not only are they charged in such a way that they have negative and positive, but when they accumulate together, as you would with a cup of water, okay, or a drop of water, think of what happens to it. If I took a little bit of water and put it on the table, it stays together. So you find forms that little droplet. If I took a drop of oil, vegetable oil, whatever, put a drop here, it kind of spreads out. Water is a little bit different, molecularly speaking. The molecules tend to adhere to each other. We have a word for that, surface tension. That surface tension is sufficiently strong that it allows water to exert a certain amount of pulling and tugging. And so when we heat water, why water's us, universal solvent in us, is even though the atmospheric conditions vary, and boys, it's another wonderful time of year to show that, because I mean, a couple of mornings ago it was ridiculously cold, and now it's relatively warm compared, it's probably effectively a 30-degree Fahrenheit difference. Yet your internal body temperature stays pretty well around normal most of the time. I think it varies by a tiny bit. That's because it takes a lot of energy to separate those water molecules. So if you had a saucepan filled with water and a saucepan filled with vegetable oil, okay, identical volumes, 2-quart saucepan, same identical saucepans. Put water in one, oil in the other, put them both on identical burners, crank the burners up to high, stuck your finger, don't do this, in there. Okay, which one's going to heat up faster? The oil. Because those molecules will immediately start to move around. They're not tethered to one another. But water has a certain degree of resistance. It takes longer to heat it. And there's all sorts of different capabilities that it has. So it's fascinating in that regard. So what water does, moreover, and a very tiny percentage of what here, plays a role in reactions. And what happens is when we look at salts, charged particles like sodium and chloride, good old table salt, and we have lots of salts in our body. So what happens with these salts, they easily dissolve in water because the water molecules can surround them. But some things when you put it into water have an impact on water. Normally, I want to get to the point where we can see that, and this is this whole idea of acidity and basicity. Okay, in the old days we used to call it acidity and alkaline. And so water has this remarkable capability of separating. I'm trying to get to I really should be writing that down. Heavens. I thought maybe it was on this PowerPoint, and it's not. So without me, I don't know if I can draw it. Maybe I can. So, assuming that this writes, look at that. Water exists, and this is what happens with all kinds of molecules. They sort of go in both directions, and a tiny percentage of water does what we call ionization. It's very tiny. So if we took water, it will separate. It's like a tug of war in that molecule that has positives and negatives. What happens is because the oxygen is pulling the electrons, every so often it wins the tug of war with one of the electrons. So it kind of looks like this. Here's the oxygen, here are the two hydrogens, and these bonds are really nothing more than a pair of electrons. And the oxygen is tugging those electrons. And every so often it wins, very rarely. So most of it stays as water, a very, very tiny amount separates into what we call partial ionization into hydrogen ions and hydroxyl. That's what makes pH. So if you take a relatively neutral substance like table salt and stick it in water, it doesn't really have an effect. It doesn't make the water more acidy or more basic. Acidity or alkalinity. But certain substances you put in there, they change it. And that's the concept of pH. It means for the partial hydrogen concentration. And it's such a small amount. If you had, and 10 to the negative 14, if you're familiar with that kind of notation. So 6 is a million, 9 is a million, 12 is a trillion. So 1 out of 100 trillion water molecules do that. Which you and I would look at each other and go, who can't? There's not 100 trillion anything, but when you talk about molecules, there's a whole lot. So you guys all have basic chemistry. Remember Avogadro's number? So in a little thimbleful of water, you've got 6.02 times 10 to this 1/3 molecules. So that, if you do the math, it's like 6 billion. And a thimbleful of water becomes charged. So you can stick an electrode in water and a light, and you do this all the time in science classes. And a light bulb will light if you have a light. Yeah, with oil it won't. If you take deionized water, if it doesn't, to pull the ions out of it, it doesn't do that. So there's charge running around. And that's going to dovetail a little bit in when Dr. Buxton starts talking about creating charged particles or free radicals in water. So that's the little introductory nugget for our purposes to water. So water is really important in that regard. And that's why in medicine, we're so cognizant of irregularities in water, of pollutants in water, of having too much or not enough water. There's a certain degree of hydration. But some folks can tolerate it a lot more easily than do not tolerate it. Let's see. So, and generically, you hear the term electrolytes. Those are salts. They're comparatively stable. You don't really damage those, I would think, with radiation. There's a whole bunch of them, but surely the most common ones are sodium chloride, good old table salt, calcium carbonate, okay, which is really probably have more of our calcium sources, potassium chloride. A lot of our table salts are impure, depending on when they get. It sort of gives salt a sort of interesting taste characteristic if you're using sea salt or pink salt or gray salt. There's a different mix of electrolytes. For those that don't know what I cook, so I'm all into that. So much food, so little time. All right, so you don't cook. It's a problem with young people. Their palates have not become expanded yet. But it's all right. I have children like that. And my oldest is going to be 36. My youngest, rather, is going to be 36. And only recently has she started to develop more of a palate for things like that. My 20-year-old granddaughter, my older of the two, I mean, it's still chicken nuggets and mac and cheese. It's okay. I don't know how she stays skinny. God bless. Okay. And then there's the pH scale, which you probably have seen before. Things that are acidic and things that are basic. And interestingly enough, our taste buds pick up a lot of this with regard to it. Let's see. And what our bodies do and another thing with the importance of water is we have the beauty of water And this is a particularly important reaction that I stress a lot in the physiology programs I teach. These are called buffers. So there's wide variations in activities we have and things that we do to create wide variation in acid and alkaline levels. How do we tolerate that? So if you're like me and had several cups of coffee this morning to get going, or a caffeinated beverage, whatever you have, there's a fair amount of acidity. So, I mean, we have the ability to deal with that acidity by something called buffers. And interestingly enough, we take water, so again, underscoring the importance of water, and mix it with another waste product, carbon dioxide, and it creates this. So the two mix together, and if you do the chemistry, H2O and CO2, you get H2CO3. And that forms what's called carbonic acid. That's a weak acid. So acidity being strong is it releases that proton, that H plus right away. But mild acids are very ambivalent. So a weak acid, and it's why they make great buffers, is they will either give up the proton or they'll hold on to it. They don't care. They're the ultimate fencer. And so what they do is they can either accept a proton or donate a proton, and that is able to compensate for variations in alkalinity and acidity. So if you go out and go to the gym and work out, you're going to develop a lot of acidity. There. So your body has the ability to compensate for that until eventually you overall compensate for that respiratorily. And what you do is you blow off excess carbon dioxide and bring in more oxygen, sort of reverse the process. That's there. So, okay. So this brings us into the different things, these sort of building blocks. So I'll give short shift to the first three and long shift to the DNA. So you have carbohydrates are just that, carbon and water, equal parts. So, and they basically, depending on the ratio is somewhat fixed with regard to this. So you have H2O and carbon. So all the formulas are carbon and water, and just how many of them there are. In our bodies, we use ones that are typically have 3 carbons, 5 carbons, 6 carbons. Not that it's a big deal. And we give them names based on it, and the names might be relevant as we go into our discussions about it. So a simple sugar, the typical sugars the sugars that you and I ingest and we use for, and then principally they're energy sugars, are 6 carbon sugars. And since we often shape them like a hexagon, we call them hexoses. And basically the one that you're most familiar with is called glucose. And then glucose is our principal energy source. And for all intents and purposes, things, whether you're having table sugar, which is a mixture of glucose and another 6 carbon sugar called fructose, or you're having malt sugars, let's say from cereal, or from milk, you're having milk sugar, which is called lactose. Eventually, our body breaks them apart, assuming you have the capability to do that. People are lactose intolerant or not. They're missing an enzyme in order to do that. And so all roads kind of use glucose, and that's really where our energy supplies come from. The 5 carbon sugars are very relevant to us because they're the composition, they're the important sugars that make up DNA and RNA. And DNA gets its name from the sugar, deoxyribose. So that's a really important constituent of DNA that you should know. So here's what the basic saccharides look like. These are the ones, and chemically our bodies fairly easily can take them apart, any combinations of them, and change them all eventually to glucose. Don't have to know the chemical formula. But so here you have a 5 carbon sugar. Each of those little points typically represents a carbon, unless there's something else there, like in this case, an oxygen, and the carbon is sort of a side chain. So this is really important. So if you look at the distinction between deoxyribose, and the name tells you what it is, it's missing an oxygen, deoxy. I took away an oxygen. So here's ribose with 2 OH groups on the bottom, and and in organic chemistry, these numbers have little positions. So you can you'll see if you go on Google something, say a chemical compound, and it has a very, very long organic chemistry name, like this. And that, and our students who do organic chemistry, they learn all of that. And 1,000 years ago, I was a chemistry major. Don't ask me how I got it. Okay, I used to know all that stuff, but that was a long time ago, like 50 years ago, so I don't remember. Don't start. See, I say 50 years ago, and you're just in here, man. So here you can see the oxygen that was deoxygenated is right over here. Other than that, they're the same. So, and they talk about the different kinds of sugars, and we put them together, and they're all sorts of interesting. By and large, biologically, what we do, with the exception of fats, these building blocks, carbohydrates, proteins, nucleic acids, which is where our focus point is, are polymers. So a monomer is an individual unit that's either identical or similar. A polymer is many of these chained together. So when we get to DNA and a little bit to RNA, they are all they're chained together. So the polymers of carbohydrates, if the single units are called monosaccharides, and sugars like table sugar are disaccharides, polysaccharides, and another name you and I give them dietarily is complex carbohydrates. And for most of us, they're the largest part of our diet. Most of us eat probably a disproportionately large amount of carbs because they're comparatively inexpensive, they're a rapid source of energy. They have their own subset of problems dietarily. So some people, diabetics for instance, can't tolerate them because they don't have enough insulin, in order to compensate or to utilize those carbs efficiently. So polysaccharides, where we ingest most of them, okay, are in the form of starches. Starches are plants, and we get a lot of our carbohydrates from plants, but our bodies are able to break them down into glucose for energy. Glycogen is a certain amount that we get from animal products if you choose to ingest those. A lot of folks these days, you see more and more people who are vegetarians and vegans and stay to a certain extent away from animal products. I have a lot of those in my family for a variety of reasons, so it gives me some opportunities to cook veg and vegan for all those things, which is kind of fun. And they aren't very soluble, they don't dissolve, but they break down fairly easily. The trouble is we can't store them terribly effectively, so we tend to flip them around and break them down and sort of convert them into fats for storage purposes, which is one of the problems with excessive carbohydrate intake. So they're all chained together in these interesting shapes that branch or don't branch or cross-link. Ones that we can't ingest, that we do ingest a lot, that we can't utilize, but ingest a lot of, are made of cellulose. Those are plant products from cell walls. So they're used for us because they provide dietary fiber, which has a lot of uses digestively, and not the least of which pulls away excess fats that we are able to excrete. So there's a lot of interesting stuff from that. Fats, the difference between fats and carbohydrates is they have a whole lot less oxygen. So they contain carbon, hydrogen, and oxygen, and occasionally phosphates. And the phosphates really become an important building block for membranes, which is something that we alter when we administer radiation. But fats are relatively resistant. So we store a lot of our energy in the form of fats called triglycerides. We have a large amount of these, but the most important of these, I would think, are phospholipids. We take something, and I'll show you it very, very briefly, not that you have to remember it. We take these compounds and really what a fat is a combination of an alcohol and an acid that come together. It's a fancy name called an ester, E-S-T-E-R, in organic chemistry. But they are a neat sort of combination that occurs, and they're extraordinarily important. So we take these triglycerides, and I'll show them to you. These are so-called storage fats. They take an alcohol. These are called sugar alcohols, called glycerol, and 3 fatty acids, and they're everywhere. So they end up with a molecule that looks a lot like this. That's a neat. You can see the abundance of carbon and hydrogen, and there's a tremendous amount. Fats are a much better energy source. They're just the problem is the waste products they create, and some folks' ability to be able to deal with it from a dietary perspective. So what we end up doing when we make a phospholipid, and they're just showing you schematically, and then they show you the illustration. And so these are examples of what they call saturated fats, because saturated means they have a maximum number of hydrogens. And they get kind of a bad rap, okay? Because people look at this and say, oh, it's butter, it's bad for you. Well, the reality is something like margarine is considerably worse. So there's a lot of dietary science behind that if you're interested. They have what are called sites of unsaturation. So if we take things and we basically create these trans fats, okay, and that's really what happens with margarine. We take things that don't have this, blow hydrogen into them, create things that are bad. So they create, when they're metabolized, they create a lot of unpleasant waste products with free radicals and errors, where these fatty acids that you might get, which I take as supplements, and a lot of folks do, or you can get them from a variety of foods. Got some wonderful salmon from IKEA from the freezer, filled with omega-3s, and immensely tasty. And I love the creamy dill sauce with it. Happy to. So here you can see, and and that's why a lot of these things with just a single site of unsaturation are very useful. Things like extra virgin olive oil qualifies that. We use a lot of avocado oil, which is another one of these relatively neutral, heart healthy kinds of things that are there. And what happens is we lop off one of those long chain fatty acids, add a phosphate, and create something called a phospholipid. And that's the foundation for living membranes. So just to give you a potpourri, just to give you sort of some basis. So cellularly, these play a very, very pivotal role. Because they naturally, fats don't bond together. They just the fatty portions have a tendency to come together naturally without being bonded. And so they tend to form, when we have the phosphate area, we call it the polar head, and we have these lipid tails, and they inherently, if you put them all together in water, voila, they tend to form membranes. And that's what living membranes are all made of, a thing called a phospholipid bilayer. So the fatty tails come together naturally. The phosphate heads point outside. Let's say if this is a cell, to the exterior or inside. And they have the remarkable ability for things to implant themselves in there, like other fatty molecules like cholesterol, which help to impart stability to the membranes, and proteins, which give functionality to the membranes. And that's really what happens in living cells. So we hear a lot about these steroids are really important for membrane stability. And because they're relatively small and fatty, they can get through those membranes in the form of like steroid hormones like testosterone or estrogen or progesterone, and a variety of others that are important biologically. So, and they're really important. We make vitamin D, which is critical for absorbing calcium, bile salts, which are critical for digesting fats, and as well as the cell membrane structure. So it's found a lot. It's not only found in animal products, but mostly found in animals. And they all kind of look like that. That's what a cholesterol molecule does. And we lop off these side chains that are here, and then all of these steroids that you hear about so much have kind of formed from this basic, the one you see there. And we don't have to deal too much with icosanoids. There's something, they're local acting fats that play a pivotal role in inflammation, blood clotting, as you can see, labor contractions, blood pressure, etc. And that's something I deal a lot with when I do my physiology courses. As we move on, proteins are all formed of something called amino acids, of which there's roughly 20 of them. In this textbook, they comment about 22. I think they all come from 20 of them. We have ones that are considered essential. When we talk about essential in science, essential means something that you must acquire that you can't make. There's 8 of them that we have to acquire through our diet. So when you hear the term complete protein, that's something, if you're eating meat or dairy products, those are by definition complete proteins. vegetable plant products are not ordinarily complete proteins. Some are. Normally you have to mix what are called legumes, which are the typical beans that are proteins, with some kind of a seed. That's why beans and rice have been a staple in a lot of diets, particularly vegetarian and vegan diets, because that combination would yield a complete protein. The one legume that is a complete protein, and why you see it so much in certain diets, particularly folks who are vegan, is tofu, soybean. The magical soybean is a complete protein. So, and there's a lot of soy in everything. Just be a label reader with all of those things. But nevertheless, a lot of our bodies are made up. These a tremendous amount of our body by weight is, or rather, are proteins. Our muscles are basically just all proteins that are there. And so they're held together. The amino acid is the monomer because they were all very, very similar. The polymeric form is chaining together of these monomers by what are called peptide bonds. So, and they make up structural proteins and transport proteins like hemoglobin and enzymes, muscle materials embedded in membranes, antibodies, which you hear so much about today vis-a-vis COVID. I just saw my middle son last night for the first time in about a month and a half because he had COVID about a month ago. Okay. And got a dose of what are called monoclonal antibodies early on in the treatment for that, and did really very, very well afterwards. So they're made up of these amino acids. What they are, they're a molecule. This is sort of the base molecule. They all look like this, and they vary, the 20 of them, by what's called this R group. And we join them together. And they form long, complex molecules. Amino acid 1, amino acid 2. The chain of amino acids is what make, when they're small chains, we call them polypeptides. When they're long chains of 100 or more, we have a name for them. Okay, so you're composed of a combination of polypeptides and proteins, all of which the sequencing of these amino acids is based strictly on the structure of your DNA. That's why it's so terribly important. So that's really the part of it. So alterations in our DNA alter the structure of our proteins. The batting order, so to speak, of these amino acids is called the primary structure. And the primary structure, depending on what it is, begins to form. And now we get the relevance of these hydrogen bonds and the impact of water. When we have these subtle magnetic attractions, they cause these chains of amino acids to form either helical structures or these sort of fan-folded structures we call pleated sheets. And they play indeed a pivotal role in our biological or internal structure. And then typically they form together. Most of the time they form together with something called a globule, which is a combination of these, but sometimes you're going to see, backtrack a little bit. These things, most abundant of all the proteins we have in our bodies, called collagen. It's a structural protein, and it's as though we took these and we almost like braided these spiral proteins together, 3 or 4 at a time, created them together, and they are inherently strong. When you braid something, you make it strong. So we used to do that in surgery. We take monofilament steel wire, which is very low reactive, and then basically fold it over and have 2 people, the doctor and whoever is the surgical assistant, and we twist it. And we create a braid out of it, which strung our wire bones together. It's kind of fun. And so that's where we get this elaborate structure. And sometimes these are the ones that are very functional that we see inside membranes and have things do things. And then this is a very elaborate structure called the quaternary structure. And this is the distinction between fibrous and globule. And so when there's the alterations in particularly pH and temperature, they can cause these proteins to unfold and lose their functionality. It's kind of what you do when you heat it, when you cook an egg. You cause the proteins to denature. And obviously it's an irreversible with temperature denaturation. Which brings us to the last part, nucleic acids, which are the most complex of these and by far and away the largest. And there are two, DNA and RNA. They have carbon, hydrogen, oxygen, nitrogen, and phosphates. All of them, okay? And the individual unit is called a nucleotide, okay? It has a nitrogenous base, 5-carbon sugar, and a phosphate group. That's critical. And depending on what the sugar is, if the sugar is deoxyribose, the long chains form DNA, deoxyribose nucleic acid. And if the sugar is ribose, it forms ribo or ribose nucleic acid RNA. The whole idea of this has to do with the backbone of the sugar and the phosphate, and that really dictates the shape of the molecule, which is so critical. So DNA, genetics, it's the coding for proteins, and it's our DNA. So as cells replicate, we replicate the DNA. The beauty of DNA is the nature of the strands, and we'll get to it. So what DNA inherently forms, if we take a repeat unit of deoxyribose, and you'll see the illustrations, and a phosphate, okay, and classically we see them in the nucleus of the cell. It's not the only location, also in something called mitochondria. Contain a deoxyribose sugar, a phosphate group, and very famously, one of these 4 nitrogenous bases. And the bases bond by what's called complementary base pairing. The bases are in the form of what are called, and most people are very familiar with the A's, the G's, the C's, and the T's, adenine, guanine, which are similar molecules and have a double ring structure, and pyrimidines, which have cytosine and thymine, which have a single ring structure. So the complementary base pairing is A's and T's, G's and C's. When they're not paired with those, by definition, that's an error in DNA, and the word for that is mutant. So here's what it looks like. And notice again, hydrogen bonds, these little dots that I will make sure I get to show you, or I may send you a little dictation about showing you hydrogen bonds separately. That basically are these subtle magnetic attractions. And the beauty of them, these hydrogen bonds, is they're sufficiently weak that it doesn't take a lot of oomph to separate them. So what DNA is, for lack of any better way to describe it, is a biological zipper. What do zippers do? We zip and we unzip. So you can open and close them. We want to replicate DNA, we open it up. And then we can replicate it from the strands that are there. When we want to turn it into proteins, we open it up and use one strand or the other to generate proteins by something called transcription and translation. Transcription means taking one of those strands of DNA and making it into RNA, and then translation is eventually making that into proteins elsewhere in the cell, something called ribosomes. So here's what they look like. So think of a spiral staircase that's uniform with regard to the width of the stairs. So if you see one of those things, you might see that, every so often you'll see them in like a loft type of an effect, going up maybe to like a master bedroom loft and a condominium. Rented a couple of those like beach ones that have that. So that's kind of what they look like. So the stair steps are equally wide, all about, though it doesn't really demonstrate it so well, but the width is such that it can accommodate one of the purines, in this case adenine, and one of the pyrimidines, in this case thymine. So the A's always go with T's, C's, and G's. But the batting order can be anything. You can have an identical batting order for any number of these. It's such an immensely long molecule. Literally billions of these base pairings that occur, all held together by hydrogen bonds. So the backbone, so to speak, is the combination of the deoxyribose and the phosphate molecule. So they form the so-called backbone that spirals on either side. And the beauty of this molecule, which makes it so unique, is it's identical. So if we take a look at that molecule, and assuming that these, they have different color top, are identical markers, they're what we call anti-parallel. So it doesn't matter which strand of DNA we use as what's called a template, a model to make proteins out of. They're the same, except one's right side up and one's upside down. That's the fancy term for that is anti-parallel. So it doesn't matter where you start here or here. But when they mutate or are damaged by radiation, now we begin to get problems. And you'll see down the road, the question is whether it's an isolated break in the DNA in just one location or if it's a double break. And single breaks are relatively easy to repair, where double breaks are. And that's something that we'll be coming to, beginning to look at those. So here's the pattern. We didn't know about this. It was the very famous Watson and Crick molecule. And if you know the story, another researcher, a lady whose name was Rosalind something, will come to me in the middle of the night, like things do. Rosalind Franklin. You didn't have to wait till the morning. When you're old, your memory. Mine does not. So what happened is she was an expert with X-ray diffraction. What that meant was she would X-ray chemicals and be able to construct the shape of the molecule from it. They couldn't do it with DNA. They were able to isolate DNA early on. There was a lot of research that stopped prior to World War II. And in World War II, then the scientists started to work toward the war effort, depending on what side or sides they were on. Then post-World War II, roughly 1948-49, the great scientists in the world began to try to identify what DNA was. The most famous were Watson and Crick in England, and a two-time Nobel Prize winner out in California named Linus Pauling, who the character Linus in the Peanuts cartoons derived his name from. So they were like the smartest people on the planet, that, and Einstein was getting older. So what she did was she just couldn't get a good X-ray, so she decided if I freeze the DNA, put it in liquid nitrogen, so effectively it stops moving, and then X-ray it, and she did just that. And then she was able to identify the uniform length, and she told everybody about that. And then Watson and Crick went about building the molecule, and unfortunately, because she was a woman, and women in science have always been mistreated, as have women in medicine. There's no excuse for it. Okay, they didn't give her credit, and she didn't really get the credit until posthumously. She worked with X-rays, and eventually she died of cancer. They didn't know about as many of the bad things that were there. But she was the one who came up with that missing piece. They knew about the equal components of adenine and thymine and guanine and cytosine, but they couldn't. And they went up in their lab, they made a Tinker Toy model. Something you probably worked with in high school. So the difference is the sugar. In RNA, the sugar is ribose, and for whatever reason, it does not form that twisted ladder molecule, the so-called right-handed or alpha double helix. It forms a single strand that does most of its activity outside the nucleus. The nucleus has tiny pores in it, called nuclear pores, that are big enough for products to go in and out, but not DNA. DNA is too big to get through the pores. And so, and interestingly, thymine happens to be replaced by another pyrimidine called uracil, though they're almost identical. There's different types of RNA, messenger RNA, which carries the genetic code with it, transfer RNA, which mates to it and is attached to amino acids in the environment around the cell, so it can pull them toward it, and ribosomal RNA, which creates ribosomes along with proteins to make the plaque for this all to occur. And it's a pretty well understood process. So when you look at these things, I don't know if
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So it's pivotal that DNA be replicated appropriately and identically at all times, because any alteration in that, we have a good capability, but a limited capability to repair. So when our capabilities of repair become limited for any number of reasons, environmental factors, people who smoke or drink excessively or are exposed to a variety of toxins or radiation, that begins to create more problems and alters our capabilities. Ultimately, it's a numbers game. We can deal with a lot of things. I smoked cigarettes for 40 years, 2 packs. 40. You guys aren't 40 years old. Think of it. Okay? Only by the grace of God, I don't have lung cancer. Unfortunately, I got bladder cancer. But understand, so there's, you're taxing your system when you're doing things like that. That's the problem. The other important nucleotide is called ATP. That's our energy resource. Basically, it's a small molecule, not a big molecule. It's not a long chain. It's individual molecules. And what it does is phosphate groups are negatively charged. When you hold them together, it's like having kitchen magnets at the same charge. You got to exert a little extra force. If you release them, they kind of pop up. Don't tell me you didn't do this as a kid. Kitchen magnets, fun to do that. Put them on a surface and try to push them around. We didn't have a lot of stuff going on, barefoot to school, uphill both ways in snow and miles. I grew up 20 minutes from New York City, believe me. It was civilized. But that's so we constantly can store energy here, and when we release it, it's a substantial amount of energy. And that's this whole idea going from ATP and ADP continuously, which we do. And that's everything for today. I will probably try to get you some additional information separately out of class about the cell cycle, and then dovetail into that when I join you back, not next week, but the week after. So thank you. And we're good. Yeah, I know you guys don't get to leave. You're like stationed here.